Hi everyone, my name is Leslie Chen. I am a 10th grader at Edward R. Murrow High School. Today, I'm going to present to you the effect of 2-hydroxybenzoic acid on Mimosa pudica lin sensitivity for the generation of electricity towards the development of a plant-based sensor for a spinal cord stimulator. It is an important role to find an alternative spinal cord stimulator for appropriate care following spinal cord injuries worldwide. This research tested the effectiveness of 2-hydroxybenzoic acid when applied to Mimosa pudica lin as a way to indicate if it will increase the efficacy of the amount of electricity produced in the plant. Mimosa pudica lin is a very useful plant for various pharmacological and biological studies relating to different human conditions. Salicylic acid is a phytohormone found in plants and plays many roles in plant physiology, and is especially responsible for the plant's response to abiotic stress. It also helps the plant's defense against pathogens. Electrophysiology is coordinated by movement, such as response to touch or any other environmental factors. Just like human beings, when we move, electricity is produced in our body. This concept also applies to this plant. This is a SpikerBox mechanism. This instrument is used to amplify and monitor the electrical activity of neurons. So when the leaf of the plant is tapped, an action potential is triggered, which causes the cells to open up, releasing water in them, changing the appearance of the plant. Hypothesis: the application of 2-hydroxybenzoic acid can result in an increase in the amount of electricity Mimosa pudica lin produces due to its ability to respond to abiotic and biotic stress. Materials and methods. For this experiment, I dissolved 0.03 grams of SA in 500 milliliter of Poland-Spring water. Since SA is poorly soluble in cold water, I mixed it on top of a hot plate at 30 degrees Celsius to properly dissolve and homogenize the solution. In my experiment, I had two groups. One group where the mimosa plant was treated with SA, and another group where the plant was treated with water only. This is a representation of my setup. On the left, you can see a SpikerBox, two cables, and the mimosa plant. The red cable is the stem cable. It is attached onto the stem of the plant, and the black grounding wire is placed inside the soil. On the left is a threshold recording of the electricity the plant is producing. At the beginning, you can see the amount of electricity increases, but towards the end, it drops drastically and flatlines. That indicates the action potential has hit the plant cell, releasing the water inside and changing the appearance of the plant. These are my results for Mimosa pudica from group 1. As you can see, hour 24, which was the first day, the amount of electricity that Mimosa pudica 1 produced was 1 millivolt. At hour 120, the amount of electricity Mimosa pudica 1 produced was at 1.8. Compared to the control group of my experiment, at hour 24, the amount of electricity they produced was 0.3, and at hour 120, which was the last day, it only produced 1 millivolt of electricity. Discussion and conclusion. The results of my experiment indicate that there is a degree of truth in my hypothesis. I hypothesize that the Mimosa pudica under the influence of salicylic will produce more electricity than the Mimosa pudica treated with water only. Biocompatibility plays a major role in this research. Although this research was driven to find whether or not Mimosa pudica lin has the ability to generate electricity, for future experiments I want to focus more on the aspects of the genes triggering the movements. These are my citations for the research. Thank you, New York Sunworks, and thank you guys for watching my presentation.